So today we'll talk to you about a very interesting story that we sort of unexpectedly came upon in the lab and tell you the story behind it. And you know, in science, most of the times you fail, and in baseball season, I've realized that to hit 3 out of 10, to hit 300, you're doing pretty well in baseball. So most of the time you do fail in science, but sometimes you're rewarded with very simple questions in the lab and get a very unexpected answer. So the topic of my talk today is to really think about what we have sort of colloquially called dark matter of cancer genomics. And what that refers to is in the fact that by and large, most of sequencing studies using massively parallel sequencing have largely focused on the exome. And that's generally about the 1 to 2% of the human genome. And to look for the oncogenic mutations and tumor suppressor alleles that have been described in the cancer research literature. One of the questions we wanted to know was, do non-coding regions of the genome contribute to cancer? And there are many difficult challenges to this question, one of which is that there are challenges both computationally of understanding these regions of the genome that are not easy to interpret. These are large intergenic regions with repetitive stretches of DNA. And what we do know something about it, and actually a lot about in terms of a lot of the basic science research that has been performed in this field is in promoters and enhancer areas. And these papers recently in the last 2 years, using systematic approaches to understand non-coding areas of the genome, will be very informative to our understanding. And so what we had come upon in the lab is actually a critical mass of whole genome DNA sequencing that we had performed with 25 whole genomes of malignant melanomas. And using that data, this data was published about a year ago, we asked a very, very simple question. And that question was, are there somatic mutations that occur more than once in these whole genomes in non-coding areas? And much to our surprise, and actually much to our surprise so much that we actually didn't believe the data at first, we found that in 17 of 19 of these melanomas that had coverage at the TERT promoter, that there were 2 specific mutations within 100 base pairs of the transcription start site. And these 2 putative somatic mutations we called C228T and C250T for the genomic coordinates and their changes from a cytidine to thymidine. And both mutations we found were mutually exclusive, and they both occurred, as you can see here, about 200 base pairs upstream from the translation start site, as designated by -124 and -146. Now, it took a long time actually to convince ourselves that these mutations were real, and I won't bore you with the details, but we essentially performed very traditional Sanger sequencing to confirm that both mutations were indeed present in the tumor, but not in the normal in melanoma samples, and then these are the matched normals for these patients. And when we did an extension cohort, what we found in the end was that 71% of the melanomas had either one of these 2 mutations. So this was an incredibly surprising result to us, and we were very surprised that this was the actual case, since this was more common than BRAF V600E, and seemed to be the most common. These 2 mutations were the most common mutations seen yet to date in melanoma. When we looked across our collection for data which we had BRAF and NRAS mutations, we found that these did co-occur with the NRAS and BRAF mutations. In the samples that we looked at. And then what we wanted to do then was determine, what was interesting about these mutations. And, there's very few moments in science when you look at something and you sort of say, wow, there's a moment of sort of eureka. And when I looked at the data and looked at the sequence, what turned out was that both of these mutations created the exact same 11 base pair sequence within the TERT promoter. And embedded within that sequence was a consensus at its binding site. So this mutation here is C250T, and this mutation here is C228T, both created a consensus at its binding sequence, and in fact created the same 11 base pair sequence within the TERT promoter. In order to test whether or not these were functional mutations, we used very well-established luciferase reporter systems. I cloned in these mutations into a portion of the TERT core promoter and then tested them in luciferase reporter assays. And in fact, tested them in a bunch of cell lines, actually that we knew had either TERT promoter mutations in them, and showed that in general, we found that each mutation conferred about a 2- to 4-fold increase in transcriptional activity from the TERT promoter. We actually didn't publish this data, but we also showed that when you actually mutagenize the site to a non-consensus ETS binding site, noted here, you actually don't increase transcription from the TERT promoter, but actually when you change it to a C228A, which is also represented by the consensus ETS binding site, you still maintain this increase in transcriptional activity from the TERT promoter. We next asked, are these TERT promoter mutations present in other cancer types? Now, I'm sure many in the audience know that TERT telomerase gene is found or is expressed or overexpressed in 90% of cancers. But for a long time, it hadn't been known why they hadn't found mutations yet in telomerase. And so we wanted to know were these promoter mutations found in other cancer types. And we made use of the Cancer Cell Line Encyclopedia, for which we had whole genome sequencing data on 150 cell lines representing a number of tumor types. And we showed that throughout these 150 cell lines, as denoted here, we could see that in bladder, thyroid, melanoma, CNS tumors, liver, kidney, mesothelioma, upper aerodigestive tract, less so in esophagus, stomach, and lung, that we could see that these promoter mutations were found at a high frequency, at least in a small number of cell lines in these lines. But then again, in these other tissues, we did not see any. And subsequent papers published after our presentation of the data showed that, and this is, I didn't cite all the papers here, but there have been about over a dozen or two papers that have then now subsequently looked for these recurrent TERT promoter mutations in a number of cancers and shown that they are highly prevalent. These two specific mutations are highly prevalent in a number of cancers. I'll draw your attention to glioblastoma, where it's present in 80 to 90% of all glioblastomas. Melanoma, as we described here, hepatocellular cancer, it's about 66%. Thyroid cancer, medulloblastoma, a number of different cancers. These are the most common mutations in these particular cancers. And this is just to depict in more pictorially that in these cancers, melanoma, GBM, bladder cancer, and hepatocellular cancer, TERT promoter mutations are the most common mutations found to date in these cancers. And they are more common than BRAF V600E, and they're among the most common point mutations observed in human cancer. So this data really suggests that non-coding regions, of the genome as important in carcinogenesis. And so what do we think really these mutations are doing? And I think that this is just a model. I don't think we've proven any of this, other than to say that that you can imagine that signaling events such as the BRAF V600E driver oncogenic mutation, EGFR amplification, other signaling events that lead to dysregulation of ETS factors, at least in the case of the TERT promoter, then allows for a binding of these ETS factors to the TERT promoter, which drives telomerase. And this is clearly, just a model and just an idea, a hypothesis of how this might work. And you can imagine that this is another model for just how non-coding mutations or promoter mutations in the human genome could act in cooperation with upstream signaling events. Just in summary, TERT promoter mutations are the most common mutations in melanoma and in many other cancer types. They're the first recurrent somatic mutations in a promoter in cancer, and they are among the most common mutations in all of cancer. These data implicate non-coding regions of the genome as important in carcinogenesis, and based on their specificity, you could say that you could imagine that TERT promoter mutations may be a useful biomarker for the detection of cancer. And I really do think that we should reconsider TERT as a therapeutic target. Thank you.
So the specificity of that motif, together with the restricted cancer types where you found this, might suggest that there is a particular family member that likes that motif and is selectively expressed in those. What that number?
Well, I think it will depend on the tissue type. I think that we know that ETV1 is dysregulated in melanomas, for instance, and so ETV1 can certainly recognize an ETS binding site. We haven't proven that that's the one that's binding at the TERT promoter. So I think that those are remaining questions that need to be.
Oh, I just wanted to ask, what about the biology? What happens in the tumors that have it if you knock it out, and if you put it in the tumors that don't have it, what happens to them? is it associated with aggressiveness or any particular biology?
Right. So, what I haven't mentioned here is that a number of papers published since in this past year have looked at that question and have shown that at least in certain cancers, I think there's data in glioblastoma and in bladder cancer that they seem to be more aggressive and that patients seem to have a lower overall survival if they have the mutation. So I think that data is still coming out and people are looking at their data sets to find these correlations.
So in those cases where there's a high frequency of these mutations, when there's also well-identified preneoplastic or benign lesions, what's the frequency of the mutations in those subtypes?
It's a great question. You know, I think what we know is that, for instance, benign melanocytic nevi, about 60–70% of them have BRAF mutations, right? So in the small numbers that I've seen of people who've published looking at melanocytic nevi, none of them have TERT promoter mutations. A group out of France published about they have a collection of benign hepatic adenomas, then cirrhotic preneoplastic nodules, and then the actual HCC, and they found none in the adenomas, and then an increased number in the cirrhotic lesions, and then 50 to 60% in your HCCs. So it seems to be a clear mechanism for transition. At least that's what the data would suggest.
Did you say that you have not identified any transcription factors as yet that bind to that region?
Correct. I mean, I think we have not specifically found the transcription factor or transcription factors that bind to that specific region in the TERT promoter. Yeah.
What else are we missing in cancer genomes in the non-coding region? Because lots of groups have looked very carefully. There are hundreds of patients who've had whole genome sequenced, and this is really the only one. I mean, this is a great find, but is it unique to this particular promoter, or are we missing other? Can you?
Well, no. I mean, I think.
Underway, the TCGA and ICGC are by January going to have 2,000 whole genomes.
Yeah.
And the analysis will be done.
Right.
I think right. I think that I would agree with that. I think that there will be other treasures to find in these non-coding regions. This telomerase is clearly a special gene, and will something come out as 90% mutated? I don't know, but there certainly will be other ones.